This DeBacco University video to go through glandular trichomes viewed with a scanning electron microscope to really give you a different perspective and a different look at these structures that you've probably seen quite a bit from a macroscopic standpoint. So this is really going to be kind of a detailed look at these basic structures. So hopefully you tune in. I'm going to read some of the information so you can really focus at looking at the quality images provided here. And those quality images were provided based on this research article here. So if you want to take a look at them or kind of look at some more of the details of the full study, you're welcome to use this citation to find this. But great images provided here, which I am encouraged to share with you. So first off, just the varieties being grown, Moby Dick and Space Queen. So they'll be abbreviated MD and SQ throughout this, just to give you a little bit of idea of kind of the abbreviation for these, because there'll be a lot of text here. So first off, start with a little macroscopic view, the inflorescence comparison, the morphology of developing inflorescence in two cannabis genotypes examined here. Again, here we're looking at Moby Dick at 3 weeks of inflorescence development. And then for B here, we've got Space Queen also at 3 weeks. You can see the different morphological differences here as well. For here, we're looking at Moby Dick on the left and Space Queen here on the right. Again, at 6 weeks. In the arrows in all of these, it show the position which the bracts were sampled for microscopic observations. So these different regions right here and these different regions right here are the areas we're going to be looking at in a very zoomed in portion. So I can kind of get appreciation for where the researchers were looking at. Now again, the comparisons, the morphology of developing inflorescence in two cannabis genotypes was examined. Looking over here, the stigmas, which is read by the ST, appear as an orange-brown coloration. The trichomes, it can be seen on the calyx, which is located right here, on inflorescence leaves of the strain for Moby Dick at 8 weeks. And we're looking at E and F, trichomes and bracts of strain Space Queen at 8 weeks here. The arrows point to the stigmas, again, that appear brown and orange as well on that variety. For letter G here, the dried inflorescence or the bud of Moby Dick after harvest. The buds has been trimmed to remove the inflorescence leaves, and the arrows point to the dried stigmas we can see right here. So again, just giving you a little bit of that macroscopic view before we now get into the detailed microscopic view. So first off, to understand the trichomes we will be looking at, there's the capitate stalked trichomes, the capitate sessile, and the bulbous trichomes. And we could kind of see here the stalked, the sessile, and the bulbous trichomes here. Looking at the bulbous being the very, very small in size comparison. The capitate sessile are basically the stalked without the stalk, so to speak, is a way to think about it, and that's looking straight down. And then if we look here, you can see the little tiny ones of the bulbous. The sessile are kind of the little bit, looks like little water droplets, and the capitate stalked are the ones here. Now description, again, the stalked are the largest, most abundant, can be seen with the naked eye, and they're a rich source of the THC and CBD. The sessile ones are smaller than their stalked trichomes, but still able to be seen and appear when the plants begin to flower. They are less potent, but still produce significant amounts of cannabinoids as well as terpenes. And the little tiny bulbous ones are the smallest of the three types. They give that glossy kind of sheen to the plant. And despite their size, they can contribute to the overall cannabinoid and terpene profile of the plant. So now let's get into some of the research images. And again, I'm going to try to provide some of the narrations here on the left so you can kind of focus on the images, and I'll try to point and highlight different aspects. So all images here are of the lower Bract surface at 3 and 6 weeks of inflorescence development. Again, we're looking at Moby Dick. A stalked-capitate trichome in the foreground here, right where the arrow is pointing, compared to the smallest bulbous ones in the background. So you can definitely see the size difference here. Looking at B and C here, looking at these two, the bulbous trichomes formed on short pedicles here, which are basically that little short supportive structure. For D, the non-glandular trichomes, as we can see with HA, there's the kind of the ones that look like points, little finger points. formed next to the bulbous, which is the one right here, and stalked-capitate trichome right here. Again, give me that comparison and really that really zoomed in look that we see here. Now what is noticed here, these little, looks like little mouths in the background are abundant stomata, and they can be seen in the raised epidermal surfaces. And if those don't know, those are the ones that are going to open and close, and those are going to regulate water for transpiration and carbon dioxide and oxygen gas exchange in the plant. So again, we're looking specifically at the trichomes, but there are other structures present. Non-glandular hairs, looking at right here, are interspaced with the stalked-capitate trichomes on a young Bract. So we can see kind of they're intermixed here as well. So kind of a cool look. All right, so there's trichome types, all images of the lower Bract surface, again, 3 to 6 weeks in development. Here we're looking at the magnified view of the stalked-capitate trichome when fully formed, head surrounded by a cuticle. So it looks like that kind of like dome-like look. It looks like it's almost like a water bag that is filled with a lot of those cannabinoids and terpenes. Here's the precursor of the stalked-capitate trichome, pointed here where the arrow is indicating, and mature stalked-capitate trichomes with fully formed heads adjacent to one another. So here we're seeing kind of a point in time, but the difference in morphological formation. Here, the mature stalked trichomes formed in large numbers and a great density. Note the presence of a small bulbous trichome right over here on the right. It's a non-glandular trichome in the background as well. Eye is the magnified view of the head of the stalked-capitate trichome with fully formed cuticles surrounding the head. So again, we're looking at those really, really zoomed in portions here of the plant. Now looking at that trichome development, let me move myself out of the way here. The capitate trichome development, again, we're looking still at Moby Dick, lower Bract surface, 3 to 6 weeks. At 3 weeks, most capitate trichomes at the tip of the Bract are sessile, but few, you can see with the arrows, have started to develop stalks. So early on in development, and we're seeing some progressing, or what they will progress to. Non-glandular trichome hairs can be seen. They look like thorns, but again, this is a microscopic view here, so they look larger. For image B, the stalked-capitate trichomes forming first along the midvein of the Bract. Abundant sessile-capitate trichomes can be seen in the background together with the non-glandular hairs, kind of mixture here, and these are the stalked ones, of course. So over here, looking at C, 6 weeks, stalked-capitate trichomes can be seen densely packed together. Again, this is kind of the goal of growers, to have a lot of those very closely packed together, high density, give me a high amount of cannabinoids and terpenes. D here, the range of stalked lengths can be seen, so they're not all the same length, with one trichome stalk in which the head has been detached, which is where we see the arrow here. So it should have the little cap on top. That one does not. And lastly, in E, the view of the edge of the Bract at 6 weeks of inflorescence development, showing greater abundance of the stalked-capitate trichomes on the lower compared to the upper Bract surface. Trichome stalk lengths are also variable here, and we see more down here on the lower surface than in that upper portion. Now looking at Space Queen, again, we're looking here at the lower Bract surface, again, 3 to 6 weeks. At 3 weeks, most capitate trichomes are at the tip of the Bract are sessile. Progressing to B, close-up view showing the mixture of the sessile and capitate trichomes with bulbous trichomes in non-glandular hairs. Those kind of points that we see here. There are no stalked-capitate trichomes in this image here. C through E, so this row right here, the pre-secretory or the sessile trichomes showing the outlines of pre-secretory disc cells. Kind of look like little mushrooms there, if you will. Progressing on to F, a range of stalked lengths have developed in the capitate trichomes, and sessile trichomes can be seen in the background. Again, a range, when they're going through and develop, they don't happen all at the same time. They happen at the same general time. That's why you get that variance there. For G at week 6, stalked capitate trichomes are densely packed together on the underside of the Bract and have formed long stalks, as we see here, and different than we saw with the previous variety Moby Dick. In H, the some stalked capitate trichomes that form at the edge of the Bract have extremely long stalks, up to 1,000 micrometers, with the arrows located right here. See the one that's definitely longer than the rest. And I is the magnified view of the capitate trichomes showing fully formed heads surrounded by the cuticle. So again, great kind of view here, makes them look larger than life, but this is actually what's going on in the kind of macroscopic view you might be more used to seeing. And kind of what does that kind of look like? Well, here we see kind of that macroscopic view of the bracts of Space Queen and Moby Dick. This one is Space Queen. This one's Moby Dick, at 6 weeks of inflorescence development with abundant trichome development. The arrows indicate and point to the approximate position where the bracts were sampled. Getting here to image part C, to quantify trichome density, the bracts were removed and placed under the dissecting microscope at 50 times magnification, and the polygon lasso tool was used to trace the outline of each segment, as we kind of see here in the black outline there. The position of each capitate trichome was marked using a pen and 5px thickness, and then counted. So what does that kind of look like? What does that data come back and say? Well, here we're looking at kind of the simulation of that data. And here we see the distribution of the capitate trichomes on the bracts. Again, Space Queen would be A and C, and Moby Dick would be B and D. This is at 3 weeks, this is at 6 weeks. So we can see the change that occurs. And the black dots represent where they're located. E looks at the density of those trichomes on the upper and lower surfaces of the bracts. So AD would be the adaxial, that would be the upper surface, and A and B would be the lower surface. So the clear is the lower, the black is the upper, here at 6 weeks again. Progressing down to F here, we can see the average trichome density of the upper bract surfaces is a function of the number of weeks of the flowering period. And then lastly, G is the density of those capitate trichomes on bracts as a function of position of sampling with the inflorescence top, middle, and bottom tier. Now what we are noticing is these different letters and getting different significance levels. We could see that here with the abdixar, which is the lower portion, tend to have a greater average trichome density. Looking at the average trichome density as well, looking at those different periods of time, and we could see this again. There's error bars indicating that there is a general progression, but occurs slowly over time and kind of peaks around that week 6 of flowering. And lastly here, looking at the Space Queen versus the Moby Dick, the average trichome density overall, the bottom, the mid, and the top. We could see Moby Dick being much more consistent, bottom, middle, and top tier, and Space Queen favoring the bottom and the mid and a little bit less on the top. Always good to compare varieties. Now we're talking at the comparison of our stock trichomes, again looking at the diameter in A and the length down here in C. The genotypes Moby Dick and Space Queen. Again, Moby Dick here in the first column, Space Queen in the second one at 3 and 6 weeks. Looking at the gland head diameter was significantly greater at 6 weeks compared to 3 weeks in both genotypes, and in Moby Dick compared to Space Queen at 6 weeks. So we're seeing a little bit of that difference in change over time. Now, the capitate trichomes length in the genotypes at 3 and 6 weeks of the flowering period here, as we can clearly see. The stalk length, again, this is the gland diameter, this is the stalk length. You can definitely see some differences there compared to the 3 weeks, compared to the 6 weeks, showing that overall development. How they actually look, well, if we turn our attention over to here, the development of those stalked capitate trichomes along the mid veins of lower bract surfaces of Cannabis genotype Moby Dick up here, and compared to Space Queen at 6 weeks of inflorescence development. So again, there are some different morphological changes. It might be a little harder to see using a loop, but from a scanning electron microscope, it makes it very distinctive. You can see much longer stalks to Space Queen compared to Moby Dick. Now we're looking at that elongation here. Again, I know this is pretty detailed, so appreciate you hanging with us. Trichome stalked elongation of capitate trichomes of Cannabis genotype for Moby Dick. Early development of stalk arising from the extension of the epidermal cells of the bract tissue. And we can see that here with the arrow right there is where it's located. Progressing on to image B, a ring of 6 raised epidermal cells demarks the early initiation of the stalk. So we're not quite 100% sure of the exact formation or the signaling that requires this, but we are able to distinctly see these morphological changes. For C here, focusing, of course, where that arrow is, this is the elongation of the stalk occurs by the expansion and upward growth of the epidermal cells, seen here in the longitudinal section through the stalk. So this is kind of the point at where things are initiating from there. Now looking at D and E, here and here, longitudinal section view of the fully formed stalk and capitate trichome showing the involvement of the epidermal and the hypodermal cells of the stalk. So we're looking physically at those structures there that are developing, that are forming this ultimately larger macroscopic structure. And F lastly looks at the cross-section view of the stalk showing the organization of 6 cells, and the interior of the cells can be seen here with the arrow. So it's kind of a unique 1, 2, 3, 4, 5, 6 here. There's kind of this unique and distinctive structure, something you may not have considered before, but hopefully developing greater appreciation for what's occurring in each of those little trichomes. The attachment of those head regions, so we're looking here shown for Moby Dick, the stalk cells here and the progression of the head, so kind of that more familiar structure that we're used to seeing. B and C show the attachment of the stalks to the glandular heads, as you can see right here, that point where that kind of cap attaches to that supportive stalk, shown in 2 dried samples, which is why they look like they have a little more texture to them. Progressing down here to the bottom row, D shows the top of the stalk showing the region here to which the glandular trichome would attach to. Here, of course, it's been removed from this. We're just kind of pop the top right off, pop the top of the muffin, so to speak, and look at the bottom. E and F is a view of the underside of the detached glandular heads showing the appearance of the underlying region, which that would be attached to the top of the trichome stalk. So it kind of shows the top portion if you kind of flipped it over and looking down at it to kind of show the texture and the cellular complexity that's occurring on these structures. Now resin extrusion here. So when we're looking at the collapse of the trichome heads shown again for genotype Moby Dick, the extrusion of resins and formation of droplets are shown again by the arrows here. A and B shows the secretion from the bulbous trichomes, and we're looking at C and E, C through E I should say, showing the secretion of the capitate trichome heads. So a little different formation, but same basic concept and idea. We're having that kind of storage organ kind of bulb out in a 3-dimensional form. F does show the bulging of the cuticle, which is showing the arrow due to the resin secretion from inside the head there, pushing it again outward. G through I here shows the secretion of the resins caused by the cuticle to develop the central depression arrows where the arrows are showing, and the trichome head to collapse. We can see again showing that region there at those different views. J through I, Q through L shows, the resin secretion causes trichome heads to stick to one another, and again by the arrows here, and the scale bars are shown. So again, I should have mentioned that before. When we're looking at these degrees of magnification, we can see the different scale bars to kind of give you a proportion there. And again, when they're getting so dense here, we're seeing that kind of merger, or that connection of those. Now, when we talk about senescence, which is kind of the formation of the harvest and kind of the, people might say the dying of or the aging of the plant. This kind of shows that senescence of those heads. So fully developed head with intact cuticle, no secretions or resins or shrinking the cuticle has occurred at this point. Arrows showing the points of ring of secretory disc cells. B is looking at this shrinking of the cuticle and shriveling of the head that has occurred, likely due to the resin secretion. C and D here, the rupture of the cuticle, right where the arrow is indicating here and here, shows the dense glandular appearance that is the resinous material underneath. E through F, the shrinkage and collapse of the cuticle on the mature glandular heads, and this is caused by the aggregation of the underlying resinous material in the surface of the heads appearing convoluted and wrinkled right where those arrows are pointing. And a close-up of the dried resinous material right here. Again, a really tight zoom in there. And H and I showing the dried convoluted appearance of the glandular heads that are completely basically senescent here. It's at the very end of the life cycle. Now look at the progression of trichome development and the senescence here. We see number of weeks in the flowering period, the trichome stalk development, the heads, the resins, and the head senescence. Where does that exactly occur? And these are looking at our genotypes again for Moby Dick and Space Queen. The proportion of the stalk trichome increases exponentially with time in the flowering periods, and it maximizes around 8 weeks. But we do see that exponential increase with that kind of peak occurring right around week 8. The process of granular heads, descence, and secretion of resins and senescence begins around week 5, again here, and progresses to week 8. So it's kind of like that last kind of part of that curve. The timing depicts here will vary by genotype, so keep that in mind, as well as environmental conditions that impose stresses on the plant, which could accelerate the process of glandular head senescence and basically degradation. So these are hopefully under, quote, ideal conditions. Now, senescent trichomes heads, occurrence of a large number of senescent capitate glandular heads on 8 weeks old bracts of the genotypes Moby Dick up here and Space Queen below. In both samples, those heads did appear to shriveled and shrunken, indicating a loss of resin had occurred. We can see that indicated right where the arrows are located. We do see slightly different structures there, but we do see that same kind of aging and that senescence of those trichome heads, which is why as growers you want to pick the peak harvest time. So looking a little bit more at the details here is looking at the trichome maturation canvas. Is the changes in translucency and the changes in the red score in trichome heads for genotype hash plant progressing from weeks 3 to week 7. So we're seeing this kind of progression over time in amount that you can kind of see through the plant. A sharp reduction in that translucency and the rays of the red score can be seen here, and that's basically what they use to indicate. Bract tissues at week 8 of flowering period, in which the reddish-brown stalked glandular heads can be seen via the arrows, and that's located right here, especially in the lower half of the bract. Looking at D, a representation of the proportion of glandular heads that are clear, which are the blue circles, translucent, which are the white circles, and red-brown, which are the red circles, using the color space that they utilized. So this really gives you the quick kind of a picture, but a quick kind of indication of what category those trichomes fall into regarding to their progression of maturation. E is looking at comparison of the appearance of the stalked-capitate trichome glandular heads under visible light on the left and under UV light on the right, showing the blue autofluorescence that's associated with glandular heads. Kind of a cool image. And then a close-up of the glandular heads and an enhanced exposure showing autofluorescence under UV light. Subaggregation of glandular heads due to the resin secretion can be seen also right here where the arrow is, indicating those heads are kind of clumping together as we saw earlier. Now, one thing I found interesting here, if you tuned in for this long, and thank you, is the post-harvest handling of our trichomes. So the effect of post-harvest handling practice on capitate trichome integrity. And this is the genotype pink kush that they used in this study. So looking at letter A up here, it's a sample that was hand-trimmed to remove the inflorescence leaves before drying, shows the majority of the trichomes intact. So hand trimming, being very careful when hand trimming. Varieties, here pink kush, those trichomes are pretty much intact or undisturbed. Looking at B, a close-up view of the trichomes in a hand-trimmed sample showing all glandular heads are intact. Not just from that kind of zoomed out version, even zooming in a little bit closer, we can see the lack of damage. Now C was a sample that was mechanically trimmed to remove the inflorescent leaves, showing a loss of the trichomes in many heads had become detached. And we can see that with the arrows. We see basically the stalk here without that nice little bulb on top. It just kind of like ends. There's not that big bulb that occurs on top. Basically, they've been removed. That's through the mechanical trimming process. D is the close-up, indicating again those mechanically trimmed. Those basically heads are broken off, especially at the edges of the bracts are the most likely to come in contact with that machinery. E and F shows the dry stalks after 5 days of drying at 50 to 55% relative humidity, show the outlines of the epidermal hypodermal cells that make up the stalk. Most glandular heads are intact with a few broken off, indicated by the arrows here. So again, this really speaks to taking the time to, doing a hand trim would be great because you're doing less damage, as long as you're careful with hand trimming, than mechanical harvester to those trichome heads. So keep that in mind. Lastly, this shows post-harvest impact on those trichomes. A comparison here under 4 cannabis genotypes have undergone post-harvest drying. So this is looking at how that drying aspect impacts the trichome development. Data is recorded from 25 trichomes with 3 replicate samples. A is looking at the genotype skunk. Glandular heads are fully formed, trichome stalks are short, and no resin is released. We can see that here with the arrow. B is the temple spice. Trichome stalks are intermediate in length and heads are releasing resin. Again, right where that arrow is indicating. A healer for letter C here. Trichomes are long. Most have secreted resin. Again, showing where that arrow is located right there. D is looking at the genotype terrapinid, where the trichome stalks are long. Many grains of trichome heads release resin. Many heads have also detached. E is the comparison of the trichome density in the 4 Cannabis genotypes based on data collected from the images. And we can see the average trichome density, and we can see the variance that occurs with the different genotypes. Standard error bars are shown there. F is looking at the comparison of the proportion of the stalked versus unstalked trichomes in the 4 genotypes. Looking at the different colorations here, the stalked representing the gray and the unstalked representing the black coloration. And then lastly, comparison of the proportion of the desiccated and senescent trichomes in the 4 genotypes. So this just speaks to, again, showing the knowing your genotype, knowing what variety you're growing, maybe growing it multiple times to get an appreciation for its characteristics, and making that selection can be very important. Because as we can see here, there's a great variability in that, and one's not necessarily better than the other. Just understanding your own variety, understanding what it produces to hopefully maximize and optimize your yield in your cannabis plants. So hope you like this kind of in-depth microscopic look at things, and we're able to tune in to the end here. It is a pretty intense look, but definitely take a look at the research article, some quality images, so you can appreciate those trichomes, hopefully on a whole new level.